Hello and welcome to Neuro Navigator. Today's topic is visual processing. Same as with the action potential series, this is going to have multiple parts. This video will focus on photoreceptors, the last one focused on eyeball anatomy, and the next one on visual pathways. Let's get started. Last video we talked about the structure of the eye and the retina. The outer segment of the retina is where a lot of light-sensitive photopigments called photoreceptors are held. Two we'll talk about here are commonly known as rods and cones, and you may even know that rods serve us very well in low light conditions, and cones help us to process color at high light levels. What you may not know is the absorption of the light by these photopigments alters the membrane potential of the rods and cones, transducing visual stimuli into an output that your brain can process. Here's how it's done. To start, we'll look at this wonderfully drawn rod. We'll break it down into an outer segment, an inner segment, and a synaptic terminal. In the outer segment up here, there are calcium and sodium permeable cGMP-gated channels. In the inner segment, there are leak potassium channels. And as usual, near the synaptic terminal and the soma are voltage-gated calcium channels. However, if you're familiar with voltage-gated calcium channels because of the concept of the action potential, this is going to start to deviate from what you know. Here, what occurs is a graded potential, not an action potential. Ultimately, the membrane potential and any release of neurotransmitter from this photoreceptor is dependent on the concentration of cGMP. In the outer segment of this photoreceptor, there are photopigments that contain a light-absorbing chromophore called retinal with an opsin protein. The surprising part of this system is that the only thing in your eye, besides an argument you might make for the lens and pupil, that responds to light is opsin. Each opsin tunes the absorption of light to particular wavelengths. In rods, this opsin-retinal combination is called rhodopsin. In cones, the retinal plus the three different types of opsins, which is S, M, and L, combined are called photoopsins. Okay, so when the light hits the receptive field of this rod, it's going to hit the rhodopsin in these discs here. You'll notice that I drew 4. There are obviously much more, but these are the discs. The opsin is going to undergo a conformational change from cis form to trans form. This will trigger the activation of about 100 G proteins in the inner disc called transduction. So the pink structure right here is the rhodopsin. The little blue structure that's attached to it is the transduction. This will then activate an enzyme called phosphodiesterase, which I'm going to shorten to PDE for my sanity and yours. PDE will catalyze the hydrolysis of cGMP to 5-GMP. As cGMP is hydrolyzed, the intracellular concentration of cGMP is reduced. GMP notably cannot activate cGMP-gated channels. Those channels will then close when the concentration of cGMP goes down, and the sodium-calcium influx will be reduced, which will hyperpolarize the photoreceptor. Remember that hyperpolarizing is when the cell is becoming more negative inside the cell than outside the cell. This means that the voltage-gated calcium channels at the synaptic terminal will also close. Now, with the voltage-gated calcium channels closed, no glutamate, which is the neurotransmitter released by this photoreceptor, will be released. This is an important mechanism. There is a lot of complex visual input that you are receiving at any given moment, and accordingly, many complicated paths have been created. This is exactly what we'll get into the next video, so follow along for more.